Cancer Research and Therapy webinar. The title of my talk today is Phytochemicals Inhibit Migration of Triple-Negative Breast Cancer Cells by Targeting Kinase Signals. As we know, breast cancer is the most commonly diagnosed cancer in females and the leading cause of cancer death globally, as shown in this representation on the left-hand side of the slide. There are various studies that have done analysis of breast tumors, and this is one of them that shows unsupervised gene expression analysis of over 1,800 breast tumors, and this analysis has segregated the samples into 5 different categories or 5 different intrinsic subtypes. There are luminal A and luminal B that are basically hormone receptor positive, and they are differentiated by the level of expression of Ki67. There is the normal-like that is similar to luminal breast cancer, and again, there are some differences in the Ki67 expression with luminal A cancers. There is HER2-enriched subtype that is a hormone-negative breast cancer, but is, of course, enriched in HER2 expression. And finally, there is the basal-like subtype, which is also known as triple-negative breast cancer, because triple-negative breast cancer, TNBC, around 70–80% of them are basal-like. And that is the focus of my talk also today. So triple-negative breast cancer, TNBC, comprises about 15% of all breast cancers. And unfortunately, because of lack of hormone receptors, estrogen or progesterone, and lack of HER2 amplification, hormonal and endocrine therapies are not feasible with TNBC. As a result, cytotoxic chemotherapy has remained the standard treatment for this subtype of breast cancer. Unfortunately, TNBC has the highest rate of metastasis and relapse, and it has the lowest 5-year survival rate among all breast cancers. We talked about metastasis of TNBC being a major problem that leads to incurable disease. Metastasis starts basically from primary tumor, but primary tumor cells grow beyond certain size and then recruit the nearby vasculature to form, through the angiogenic process, to form these capillaries within tumor. And this allows cancer cells to migrate through the tumor and access the vasculature, travel in the circulation, and basically extravasate and form secondary or metastasis tumors basically in other organs. And this is what kills cancer patients, over 90% of cancer patients. The images here show the process of cell migration. This is a critical process in
sorry to interrupt you.
Pardon me?
I'm sorry to interrupt you. So we can see the slides are moving.
You can or you cannot?
We cannot. So we just see only the first slide itself.
Okay. All right. Can you see the slides here?
No.
Can you see the slide now?
Yes, I do.
Okay, so I'll probably continue from here. Did you hear what I was saying on the video, or I should start from scratch?
Actually, I can hear the voice, but the slides are not moving.
Okay, so hopefully now it will be good, right? So you can see the slides moving?
Yes, its slides are moving.
All right, so
I start from this slide that we saw in the pre-recorded video. So here we are looking at the images of cell migration. The bottom left one is basically migration of melanoma cells through skin to reach a vessel, and the process is basically, as shown, is a collective migration of cancer cells. And on the right-hand side, we are looking at an in vitro assay where breast cancer cells are migrating collectively through a collagen matrix. And therefore, this emphasizes the importance of this process, migration and invasion through the stroma in formation of, in initiating the process of metastasis. And this process is also important in secondary sites where the extravasated cells basically need to migrate and invade through the stroma of the host tissue and then survive that environment, grow and form secondary metastases. So one of the issues that we have been thinking about and trying to address is this question that chemotherapies and targeted therapies primarily aim at eliminating cancer cells, and they are not designed to inhibit cell migration. And because of the heterogeneity in the composition of tumor cells, these therapies basically do not kill 100% of cells, and therefore after tumor relapse or even surviving cancer cells will have the opportunity to basically detach from the primary tumor and migrate, invade through the stroma to access vasculature and basically initiate the process of metastasis. So we thought to look into this question and whether or not it would be feasible to find compounds that have effects against migration of TMBC cells. So what we did was basically doing a survey in the literature, specifically focusing on phytochemicals, because we thought these phytochemicals or naturally occurring compounds can be used as both neoadjuvant and adjuvant treatments. And because of, generally because they are less toxic, significantly less toxic than chemotherapeutics that are used in clinics. So we thought these compounds can be given to patients basically on a continuous basis before and after surgery, and for years that hopefully to come after the surgery. So the phytochemicals or naturally occurring compounds are known to present a lot of anti-cancer properties. We sort of narrowed down our search to those compounds that had shown some effects in vitro assays or in vivo assays, basically, on migration of some cancer cells, not necessarily breast cancer or TMBC. So our goal was to see if we can get some of these compounds and look at their activities against migration, invasion, and metastasis of TMBC cells. And that reduced our search to these 11 compounds that showed some efficacy, basically, against migration of cancer cells, either in vivo or in vitro. The next step that we did was using a high throughput cell migration assay that we have developed in my lab and looked at anti-migratory effects of these compounds. The assay is very simple. We print a drop of a hydrogel in each well of a 96 well plate. We use a robotic liquid handler to center this droplet in the middle of each well. And then we add the suspension of cancer cells. Presence of that hydrogel drop prevents cancer cells from adhering to the area underneath that droplet. So we form this donut shape or ring shape, basically, layer of adhered cells. And then the hydrogel over time dissolves and allows the cancer cells basically to migrate from that layer into the available space in the middle of the well and eventually close this gap. And then we have developed some automated image processing techniques to compute basically the area of this gap before the migration starts and when we stop the experiment, basically. And based on these two areas, we define a simple inhibition of migration index here. If a compound is very effective against migration, the size of this area will be the same in pre-migration and post-migration, and therefore inhibition of migration will be almost 1 or 100%. And in practice, this index will be somewhere between 0 and 1. So using this, we started screening these 11 compounds against 2 triple-negative breast cancer cells. The first one is MDMB231 cells that I'm showing here a typical experiment. This is a dose-dependent treatment of these cells with fisetin. As you can see, there's a dose-dependent inhibition of migration. The higher the concentration, of course, the larger this gap, meaning that there's a greater anti-migration inhibition. And we repeated this experiment also with MDMB157 TNBC cells. One important note here is that we only use those concentrations that were not toxic. So basically, we performed some toxicity assay and only use those concentrations that gave us a cell viability of over 90%. So in this case, it was up to 200 micromolar of fisetin. After completing the screening with these 2 cell lines and 11 compounds, these are basically the results of inhibition of migration. All compounds have some effectiveness against migration of these 2 cell lines, but fisetin and quercetin sort of stood out as the most effective ones against both. In both cases, we have over 80% of inhibition of migration using these compounds. At this point, we further narrowed down our search of these compounds to fisetin and quercetin. And the question that we asked was whether these 2 compounds can be effective against migration of a larger number of TNBC cells. We selected a panel of 9 triple-negative breast cancer cell lines and performed this screening again, this time with fisetin and quercetin only. The results are shown in the graphs. The bars to the left show the highest inhibition, and as we move away to the right, those have the lowest inhibition. With both compounds, we can see again there's some effectiveness of about 20 to 80% against migration of these cell lines. And very interestingly, both of these compounds are most effective against TNBC cells that have a mesenchymal morphology or mesenchymal phenotype. That those are the ones to the left of the graphs, and usually the mesenchymal looking cancer cells have more migratory and invasive behavior than basal-like and other subtypes. So therefore, this simple screening got us to the point where we can now confidently say that fisetin and quercetin have effects against migration of triple-negative breast cancer cells, at least in vitro. The next question was whether these compounds can also be effective against invasiveness of TNBC cells. So the previous experiments were all done in cell monolayer experiments. These are 2D experiments. This time we encapsulated spheroids or 3-dimensional cultures of 157 and BT549 TNBC cells in collagen. So the top left image here is showing basically this tumor microtissue that we have formed. The spheroids is sitting within a collagen matrix. This is again for presentation purpose only. Usually we do this in a multi-well plate format, which is shown in the bottom picture here, bottom left picture. The spheroid is within collagen matrix. On the right-hand side, we are showing the result of these experiments where without any treatment, the vehicle control cells basically start invading through the collagen matrix, and by day 3, we have a significant invasion, whereas both fisetin treatment of MDMB157 and BT549 cells almost completely inhibited invasiveness of these cells in collagen. We repeated this also with quercetin, and the results were identical. Therefore, these compounds have also anti-invasive properties in 3-dimensional cultures. The next question was whether these compounds also have effects against metastasis of TNBC cells. For this purpose, we used zebrafish assay. We injected about 100 TNBC cells that were fluorescently labeled red into the yolk sac of 2-day post-fertilization zebrafish. And then the zebrafish vasculature is also fluorescent green. We look at the number of cells that metastasize through the vasculature toward the tail region. And if we look at the vehicle control condition, here we are showing results with MDMB157 cells. We can see a significant number of the cells have migrated basically toward the tail region. You can see it in this red labeled image. And then we can also see with the yellow color, which is superimposition of red and green color here, showing that the cells are metastasizing toward the tail region. On the other hand, with the Fisetin-treated group, we have significantly reduced number of TNBC cells that are metastasizing toward the tail region. We did quantify the fish that showed metastasis in the treated group. About 46% of the cells of the fish had metastasis versus in vehicle control that had about 70% of the fish metastasis. And then when we counted the number of cells that had metastasized in the treatment group, we had significantly less compared to the vehicle control group. Here we basically counted fish that had at least 5 cells in the tail region as the fish with high metastasis. And based on this experiment, we can see that the Fisetin also reduces metastasis of TNBC cells toward the tail region in zebrafish. We repeated this experiment also with BT549. Results were fairly comparable. The compound was more effective against 157 rather than BT549. Here, with this cell line, we had to begin with fewer number of fish that showed metastasis. We had about 45%, and treatment reduced it by about 12%. And the number of cells that also extravasated toward the tail region reduced. We have more variation here in the treated group, but the median of this group is at 0 compared to the vehicle control group. Therefore, the conclusion of these assays, monolayer cell migration experiment, 3D cell invasion assay, and zebrafish assay is that Fisetin, and repeating, of course, this assay with quercetin, they both have effects against migration, invasion, and metastasis of TNBC cells. The next question we asked was, what is the mechanism of action of these two compounds? So what we did here was using a human phosphokinase array. This is an array that contains activities or shows activities of 43 different human protein kinases. And each protein kinase is identified by a pair of dots and the darker the dot, basically, the higher the activity of that particular protein kinase. So we first looked at the basal activities of these protein kinases in all 9 TNBC cells. And this assay segregated basically activities into 4 different clusters. The bottom cluster here are the proteins with very high activity. Then there's a top cluster with moderately high activity, and the middle 2 clusters with fairly low activities of these protein kinases. Interestingly, when we look at the proteins in the bottom cluster which have high activity, almost all of them, except for p53, basically connect with PI3K-AKT pathway. They are either part of the PI3K-AKT pathway or are substrates for this pathway. And in addition, there is the p53 mutant that also has high activity in TNBC cells. And this is again consistent with what we know about TNBC, that a lot of TNBC tumors have high PI3K-AKT basal activity. So this was a good piece of data at the beginning to make sure that our results are consistent with what is known about molecular signaling in TNBC. Then we treated the cells basically with Fisetin, and on the top left, we are looking at the phosphokinase array again from the vehicle control and Fisetin treatment. We quantified these dot plot arrays, and 7 different proteins showed significant differences in activities under treatment. And I've listed all 7 here. And out of these 7, PRAS4D, Vinc1, AKT, GSK alpha, all 4 of them are related to PI3K-AKT pathway, meaning that there is significant activity of these compounds, fisetin and quercetin, against this pathway. We can also see that in the heat map that almost in all 9 cell lines, the activities of these proteins have significantly reduced, as shown by the purple color in the heat map here. Quercetin gave us a fairly similar result, as I mentioned. Again, we can see in the heat map here that these proteins in the PI3K-AKT pathway have significant downregulation in their activities. So therefore, the conclusion here was most likely fisetin and quercetin interfere with signaling of PI3K-AKT pathway to inhibit migration, invasion, and metastasis of TNBC cells. To ensure this is the case, we used a specific inhibitor of PI3K-AKT pathway called GSK 1059-615. This is a dual inhibitor of PI3K-mTOR, and treated 4 different TNBC cells that had high migration in 2D assays and in collagen invasion assay. We treated these doses dependently with GSK, and what we observed was by increasing the dose of the inhibitor, basically, we have more inhibition of migration, which again emphasizes the role of PI3K-AKT pathway in migration of TNBC cells. To ensure this was due to on-target effect of GSK, we looked at phosphorylation of AKT, and as we can see, by increase in the concentration of the compound in all 4, there's downregulation, or there is less activity of AKT, which is also consistent with inhibition, with increased inhibition of migration. So the conclusions of this study was these 2 compounds, fisetin and quercetin, have broad and significant activities against migration of TNBC cells in 2D assay, invasion of TNBC cells in 3D assay, and also metastasis in zebrafish. These effects were generated primarily by interference of these compounds with PI3K-AKT signaling pathway. And another important conclusion here is in clinical settings, especially these days, the use of combination drug treatment has become more and more attractive to prevent or to block basically resistance to treatment with a single drug. However, we know that these compounds, these combination treatments are usually also very toxic. They generate a lot of off-target toxicities. With phytochemicals, as we saw in the heat map of human phosphokinase arrays, these compounds generate a lot of effects against different signaling pathways, such as PI3K-AKT. We have analyzed, for example, MAP kinase ERK pathway. We have seen also downregulation of activities of that pathway. We think that these compounds can have broader effects against multiple signaling pathways that are usually active in breast cancer and other cancers without generating extreme cytotoxicity that we see from combination treatment. Therefore, this is of course something that must be tested in vivo, but we think that these compounds can potentially be used to reduce activities of multiple oncogenic pathways simultaneously. The results have been published recently in 2 different papers that I've listed in BMC Cancer Integrated Biology, and we acknowledge the funding from NIH and NSF for this project, and also the former trainees that contributed to the completion of this project. Thank you for your attention, and I'd be happy to answer any questions.
Thank you very much, Dr. Hossein Tavana, for the wonderful presentation. So now the session is for questions, is open for questions. Any questions from the participants? Any questions, you can write in the chat box, or else you can directly ask Dr.